Okay, I think we're going to get started. Welcome, everybody. It's a great pleasure to introduce today's speaker, who is Professor Moshe Oren. He actually needs very little introduction to our audience today, but I'm happy to share a few words. Moshe received his college training at Tel Aviv University, followed by a PhD at the Weizmann Institute, to which he later on returned for essentially all of his faculty career. He carried out postdoctoral research at Princeton and SUNY Stony Brook, where, working in the laboratory of Arnie Levine, he carried out some of the most important work in recent molecular oncology, where he cloned the P53 gene. And in so doing and studying early P53, provided many of the crucial tools for a lot of the research that's been carried out worldwide on this extremely important gene in cancer. Subsequently, he established his independent lab at Weizmann, where he carried out a number of the critical sets of experiments that established P53 as one of the absolute prototype tumor suppressor genes in cancer, including demonstrating that it could suppress oncogenic transformation of cultured cancer cells or cultured cells. It could induce apoptosis in cancer. It could cause cell cycle arrest, all of the cardinal features that we typically think of when we consider P53 as a tumor suppressor gene. And Moshe played a huge role in also identifying mechanisms of action, which included its participation as a target of MDM2 for negative regulation of P53 via protein degradation of P53, numerous insights involving gain of function mutants of P53, transcriptional targets of P53, and very interesting activities involving the contributions of cancer-associated mutants in P53 to inflammation-associated cancers. As I mentioned, he spent essentially his entire faculty career at Weizmann Institute running his laboratory. He became the dean of the Faculty of Biology there, and since 2015 has been the director of the Morose Integrated Cancer Center at the Weizmann Institute. He's been on numerous editorial boards. He's received a very long list of distinguished honors, including recently, not even that recently, the induction into the National Academy of Medicine, National Academy of Science. It is our enormous pleasure and gratitude to Moshe for speaking today. And the title of his talk is Mutant P53 Gain of Function in Cancer. Before Moshe starts, I just want to mention in advance, if you have questions, we ask you to please type them into the Q&A function here in Zoom. We're not going to be doing the question and answer out loud, so please type them in, and then Moshe will reply to them at the end of his talk. Okay, Moshe.
Okay, thank you, David, for the very, very kind introduction, also for the invitation to speak in this distinguished forum. Okay, let's get started. Just as a reminder, some very basic facts about P53. So P53 is a potent tumor suppressor, and it's encoded by the gene that is called TP53 in the human genome. And it makes its living primarily as a sequence-specific transcription factor. This is wild-type P53, the tumor suppressor. But as I think everybody knows, P53 is often mutated in cancer. And in fact, the TP53 gene is the most frequently mutated gene in human cancer. We're talking about half of all cases of human cancer carry P53 mutations. Depends on who is counting and how, but that's about where we are. And it's more than any other gene that is involved in human cancer. And in about 70% of those cases, the mutations in the TP53 gene, which from now on I'll just call P53, are missense mutations. So we have a single amino acid substitution, single nucleotide, single amino acid substitution. And the majority of the missense mutations occur within the DNA binding domain of P53. And some of those mutations are more frequent than others and are referred to as hotspot mutations. I think the next slide gives you some kind of very simplistic cartoon view. So at the bottom, you see the simplified view of P53. It looks like a very typical transcription factor. It has actually two transactivation domains, a DNA binding domain, which occupies about half of the protein, the central part of the protein, and has nuclear localization signals, and also has tetramerization or oligomerization domain, because it works as a tetramer when it binds to DNA and transactivates genes. Now, you can see that the majority of the mutations, this is from the YARC database, are within the DNA binding domain. And you can note that there are a few amino acids which are more frequently mutated than others. And I'll draw your attention to 175 and 273 because we are going to meet them again very soon. So what do P53 mutations do in cancer? So first and foremost, they abolish the tumor suppressor activities of wild-type P53. And there's no question that this is the main reason or the primary reason why they occur and why they are selected for in cancer. But in addition, however, some missense mutations, sometimes, I think I wrote it may, I think the evidence is already better than just may, sometimes endow the protein with a novel gain of function, the mutant protein, single amino acid substitution, with gain of function oncogenic activities, which promote cancer instead of suppressing it. And this has been studied and demonstrated primarily with the hotspot mutations, with relatively little work done on the hundreds of less common mutations. And mutant P53 gain of function, mechanistically, maybe I should qualify it first. Wild-type P53 transactivates genes via sequence-specific binding to its recognition elements within the DNA. Practically all the DNA domain mutations that occur in cancer, definitely the hotspot ones, abolish the recognition of those sequences by P53. So the mutant P53 protein does not bind anymore to consensus wild-type P53 binding site, but nevertheless, it does get to chromatin, and it gets to it by piggyback mechanism. So mutant P53 gain of function is believed to be exerted via modulation of the cell transcriptome. And I say believed because there are quite a lot of proposed mechanisms for mutant P53 gain of function. Most of them converge into this statement, but there are also exceptions, one of which I will discuss during my presentation. So mostly it's believed to work via interaction of mutant P53 with a variety of other transcription factors. These are just two examples, NF-kappa B and ETS2, but the list is more, includes close to 10 different transcription factors that bind directly to DNA. Mutant P53 piggybacks on them, gets to the chromatin, and helps or modulates the ability of those transcription factors to turn on their target genes, or very often a subset of their target genes. So typically, mutant P53 gain of function relies on robust accumulation of the mutant P53 protein in cancer cells, as is often seen in P53 mutated tumors. So a common observation, as you see here, in tumors that have P53 missense mutations, is that the P53 protein, the mutant P53 protein, accumulates at high levels. Sometimes it could be as high as 100-fold more than the normal P53 levels in the normal tissue surrounding the tumor. And this abundance of mutant P53 seems to be critical for its ability to exert gain of function activities. And this is just an example from a head and neck cancer nodule. And you can see within the cancer nodule, very intense staining of P53, mutant P53, mostly in the nucleus. And in the adjoining tissue, there's practically no detectable staining because wild-type P53 is usually expressed at very low levels and is hardly, if at all, detected by standard immunohistochemistry procedures. So when we talk about P53 mutants in cancer, altogether, the record includes close to 1,000 different P53 mutations that have been recorded in tumors, but some of them probably are just passengers, while some of them, the more common ones, are most probably contributing actively to cancer. And when we look at the common P53 mutations, we can divide them roughly into two groups. One type of mutations is DNA contact mutations. These mutations carry single amino acid substitutions in residues that contact directly with the DNA and determine the secret-specific binding of the wild-type P53. And typically, those mutations simply abolish the recognition of these target sites by the mutant P53, but they don't cause a substantial change in the structure of the protein. So if one looks at it superficially, the DNA conjugate mutants look pretty much similar structurally, conformationally to wild-type P53. Now, the other class is the structural conformational mutants, and we have more of those. And in those mutations, the structure of the DNA binding domain and the structure actually of the entire protein is distorted, misfolded. Regions that are normally buried within the DNA binding domain become exposed, and the protein is very much kind of a neomorph, looking different than wild-type P53. And now the prototypes of those two classes, R273 is the classical DNA conjugate mutant, R175, which is very structurally distorted, is the prototype of the structural proteins. And if you recall, there's actually the three very, very hot spots are 273, 175, and 248. 248 is a DNA conjugate mutant, but actually the mutation also causes some disruption of the structure of the protein, and it's often sort of in between, maybe closer to 273. Okay, so that's in general. So I'm going to share with you two stories, both still unpublished, by two outstanding members of the lab. The first story is from Ori Hassin, who is an MD who is now doing a PhD in the lab. And Ori asked the obvious question, are all P53 mutants equal, or are some mutants more equal than others? And just to remind you, we already discussed it. And here specifically, does having a 273 histidine mutation or R175 histidine mutation, these are the most common mutations in these particular hotspots, does it make a difference whether you have this mutation or that mutation? And he looked specifically at colorectal cancer. And right away, when he looked at a database of, this is TCGA, together with CPTAC, so to get a larger number of samples, what he noted right away is if one looks at tumors that have been sequenced while they were at stage 1 or 2, early stage, 175 mutations were substantially more abundant than 273. So this is the ratio 175, 273. So overall, colorectal cancer has more 175 mutations than 273. However, if you now go to late stage tumors, the ratio changes, and now you see 273 mutations more abundant than 175 mutations, suggesting that having, of course, very, at this stage, very speculative, that having a 273 mutation may increase the likelihood that an early stage cancer will transit to a late stage and progress and therefore have a worse outcome. And this caused Ori to say, okay, let's follow this and understand what's going on there. And okay, so just incidentally, even if one looks at late stage tumors, and this is based on, this is a database of stage 3, 4, mostly stage 4 patients from Memorial Sloan Kettering. And if one looks at metastasis at uncommon sites, right? Brain, bone, pelvis, peritoneum, and gynecological organs of colorectal cancer. If one looks at, compares truncation mutations, which in fact are sort of null mutations, about 30%, let's say, of the P53 mutations are truncating, null mutations, no missense protein made, R175 or 273. 273 significantly more associated with uncommon metastasis, which usually are associated with worse outcome. And if one goes and just does the obvious Kaplan-Meier curve and looks, in this case, at this two cohorts of patients, and we needed a larger number because you want to get significant numbers of cases with each of those mutations. You can see that when analyzed at an early stage, stage 1 or stage 2, having a 273 mutation bears a significantly reduced disease-specific survival compared to having a 175 mutation. So having a 273 mutation is bad. 175 mutation is not as bad. Okay. So Ori went out to understand what is the difference between those two mutations. And to that end, he took SW480 cells, which is a colorectal cancer cell line carrying endogenously these two mutants, 273H, and this one, which is not a hotspot mutant and is believed, and now we are testing it directly, not to have any oncogenic activity. Actually, it looks like it has even residual wild-type P53 activity, but that's the one that we care about. So we took cells which have been knocked out for the endogenous P53, and he introduced back into those cells either the 273 mutant, the one that these cells carry normally, or the 175 mutant. And this just shows you that expression levels were similar in both of them. Let's see. Okay, fine. And then he asked, okay. Then he did RNA-seq analysis to ask what is the transcriptional impact of each of those two mutations. And somewhat surprisingly, in this case, what he found is that whereas the 273H mutant caused massive changes in the transcriptome of the cells, the R175 mutant induced only a moderate set of changes. And sorry, where are we going? Okay. And this is when he did this heat map shows the comparison. And what you can see is that the parental knockout cells and the 175 don't differ very much in their gene expression pattern. However, 273 is very different. Of course, this is just differentially expressed genes. This is not the entire transcriptome. The other genes were not significantly different. And altogether, when he looked at this, he could identify a set of 140 genes, which are selectively upregulated by 273 and not R175 when compared to the knockout parental cells. And this group of 140 genes selectively upregulated by the 273 mutation, we already defined as the R273 signature. Okay, so that's one cell line. Of course, we want to see whether this is a more common observation. So Ori took a number of approaches to test or to demonstrate that this is indeed the case. So this is one such experiment. This is another colorectal cancer cell line, RKO, which happens to carry wild-type P53. Again, wild-type P53 was knocked out by CRISPR in those cells, and Ori introduced into those P53 null cells, either 175 or 273. And he looked at genes from the signature, and as you can see, whereas 175 does not induce those genes above the level in the knockout, 273 induces those genes significantly and selectively. So that's one more cell line. So then he looked at, okay, so before we go to just mechanistically, an obvious question is this a difference in the ability of those two mutants to bind to the chromatin of the target genes? And as shown here, this is indeed the case, at least for a subset of genes that Ori looked into more carefully. So these are ApoE and integrin A7 are two genes which belong to the 140 gene 273 signature. And this is a chromatin immunoprecipitation PCR assay. And whereas R175 does not bind to the chromatin of this gene or that gene, 273 binds significantly to the chromatin of both those genes. So for whatever mechanism, which we still are trying to figure out, 273 is recruited to those sites, 175 is not recruited. Presumably that would mean that there must be a specific interaction between 273 and not 175 with a transcription factor or a DNA binding protein that is selectively associated with those genes, something that we are in the process of figuring out, of course. Okay, so back to the generalization. So to see if this is a more 273 signature indeed is associated with the 273 mutant more broadly. So already looked at the CCLE cell line, the cancer cell line encyclopedia, cancer cell line encyclopedia. And again, looking at the expression of the 273 signature, and the 273 signature of those 140 genes were preferentially significantly higher in this ensemble of cell lines containing 273 mutations, as compared to truncating mutations. You don't see 175 here because CCLE contains only 3 colorectal cancer cell lines that have 175 mutations. And that's interesting because this mutation, incidentally, but we are beginning to appreciate it more and more, 175 is the most frequent overall P53 mutation in colorectal cancer, but apparently it's extremely hard to establish cell lines with carrying these mutations, whereas 273 cancers easily develop into cell lines. And in fact, there are altogether only 3 listed cell lines in the CCLE and in the literature that have a 175 mutation. We tried to get all of them, and it turns out that also the labs that have been able to grow them find them extremely hard to grow and very, very tricky. So there's something there in the 273 which makes the cell line much more happy out of its normal context, and also makes tumors that have this mutation more aggressive and more metastatic. Might be coincidence, but probably not. Okay, so now this is still cell lines. So now looking into real tumors. So this is going into the TCGA colorectal cancer cohort. And if we run the 273 signature, this is a gene set enrichment analysis. 273 signature run, in this case, comparing tumors that have R175 mutations to tumors that have 273 mutations. We see that the 273 signature is strongly and very significantly associated with tumors that carry 273 mutations. And same thing if one compares truncating mutations versus 273 mutations, these would be the proxy of P53 null. And again, very significant, highly significant 273 signature is enriched in those tumors that carry the 273 mutation. So it's not just a kind of tissue culture cell line artifact. This is something which corresponds to what is different between these types of tumors also in patients. And again, if one now takes, previously I showed you that if one does a Kaplan-Meier curve comparing the 273 mutation to the 175 mutation, 273 mutation in colorectal cancer has the worst outcome. If now instead just takes the signature rather than the mutations, and this is the lower quartile, the upper quartile of the signature, taking all tumors regardless of what mutations they carry. And I should say, qualify, this signature is also seen in other mutants. It's not only in the 273, and not all 273 mutants carry it. So it's not a perfect overlap. But if one now looks at the signature instead of the actual mutation, you get the same pattern, having high signature of, having a high 273 signature is a harbinger of bad outcome. So these genes, having high levels of these genes is bad for the patient, and the 273 mutants specialize in upregulating those genes in colorectal cancer. Okay, so what are those genes? And of course, when one does this gene ontology enrichment, in this case using Metascape, some of the pathways that were enriched in the 273 signature, you can see some other interesting ones, but some of the ones that were enriched significantly were Rho signal transduction CDC42 pathway, which are pathways and genes comprising those pathways which are associated, proteins actually, right, that are associated with cytoskeleton dynamics, suggesting that maybe 273 changes the properties of the cytoskeleton and makes the cells more motile and more migratory, something that one would associate intuitively with a more aggressive and more metastatic phenotype. And just to go deeper into that, Ori did, in fact, this is with the help of the Ashish Noranha from the lab of Yossi Yarden, our collaborator, did an analysis of the activation state of CDC42, RAC1, and ROA, three proteins that are part of this RO signaling pathway. And as you can see, CDC42 and RAC1 are significantly more activated when you have a 273 mutation compared to R175. ROA actually is not changed. So there is an inhibitor that inhibits CDC42 and RAC1 and not ROA. And where is that? Okay, we'll go back to that. And this inhibitor is called MBQ167. And if Ori takes a cell line that expresses the cells that express 273 and measures their migration, in the absence or presence of the inhibitor, this inhibitor completely wipes out the migration. So getting back, this introduces the previous, the one that I showed you previously. So again, prediction that these cells should be more migratory. In fact, this is the case. So in this case, we are looking at transfer migration assays or transfer invasion assays across a matrigel coated membrane. And this is, the upper one is migration, the lower one is invasion. And in both cases, you can see that very robustly 273 increases both the migration and the invasiveness of these SW480 colorectal cancer cells, whereas 175 doesn't do anything compared to the knockout, nothing above the background, the basal level. So indeed, yes, this mutation causes enhancement of pathways that promote cytoskeleton dynamics, promote migration, promote invasion, and this migration is inhibited by an inhibitor of a dual RAC1 CDC42 inhibitor. Does this also translate into metastasis, at least experimentally, as one would predict from what one sees in the human patients? So Ori did two types of experiment. One is the kind of quick and dirty experiment of tail vein injection, which doesn't really measure metastasis, but it measures the ability to colonize in the lung. And as you can see here, so these are the cells are GFP labeled, and these are the lungs of mice tail vein injected with SW480 cells expressing 175H or 273H. And you can clearly see that 273H causes more metastasis and also larger metastasis or larger lung colonies. This is a more representative, I'd say, more close to physiology assay. So in this case, Ori injected these cells intrathecally to syngeneic mice, not syngeneic, sorry, to immunocompromised mice, and monitored the appearance of metastasis in different organs. In this case, when he injected 175, even though the numbers are quite small yet, with 175, we didn't see any metastasis in the liver, lung, or peritoneum. 273, we saw metastasis in both liver, lung, and also one in peritoneal. So indeed, this mutation also enhances metastatic capacity. So the conclusion from this initially is that P53 mutations are not just P53 mutations, but it really matters which mutation there is. And just without getting into further details, it also depends very much in which context we are looking at. So 273 mutations bear worse outcome in colorectal cancer, but this is not necessarily true for every type of cancer and other cell types in which this is not the story. And this very much depends on the biology of the tumor and what is driving the malignant behavior of this tumor. And I think we can also see a glimpse of it here, getting back to colorectal cancer. So colorectal cancer was a few years ago, an international consortium divided colorectal cancer into four consensus molecular signature subtypes, CMS 1 to 4. And the CMS4 is the one that has the worst outcome. It is associated with immune inflammation, stromal infiltration, TGF-beta activation, and has the worst outcome. CMS2 is the canonical colorectal cancer, Wnt activation, MYC activation, usually immune desert, and these tumors usually have better outcome than these tumors. Now, if Ori now takes the 140 gene 273 signature and runs it against the transcriptional signatures that were used to define each of those 4 molecular subtypes, what you can see is that the 273 signature is highly correlated with the CMS4 signature, meaning that many of the genes that are defining this signature also define this signature. And we also see a significant overlap with CMS1, which is a small group, which has a distinct familial background. It's a different story, but it has no correlation at all. In fact, a slightly negative correlation with CMS2, also not much with CMS3. And this can be also shown in the gene set enrichment analysis here. So just to remind you, this will tell us that 273, okay. How about 175? So 175 correlates nicely with that CMS2. And as you can see, if we take 175 tumors compared to truncate or to 273, the 175 tumors are enriched in the CMS2 signature, telling us that each of those 2 types of P53 mutants is selectively or preferentially associated with a different type of colorectal cancer, different type driven by different mechanisms and having different clinical features and clinical outcome. Now, this could be only a correlation, but what I hope that from what I showed you in the in vitro and mouse experiments, we believe that this is not just a mere correlation, but in fact, at least in the case of 273, 273 is the driver of this behavior, or at least one of the drivers that dictate this particular biological behavior, these features, which are eventually correlated with the worse outcome. So I think, okay, this is a reminder again, but now we go to the conclusions of this first story. So different P50 hotspot mutations have a different impact in colorectal cancer, and different mutations orchestrate different transcriptional programs in colorectal cancer. And we propose that different P53 mutations might be selectively advantageous in different stages of colorectal progression by driving the activation of non-identical oncogenic pathways. And just parenthetically, we believe that 175 mutations, in fact, are advantageous in early stages in initiating colorectal cancer, because they are associated with, and this is something that we have not explored in depth because still to be done, because they are associated, at least by the CMS categories, they're associated with the classical Wnt-driven high MYC type of cancer, which is known to be, these are the events that really initiate the canonical colorectal cancer. So we believe that 175 mutations are more significant in initiating the disease. What once a patient acquires a 273 mutation, this will greatly increase the likelihood that the disease will become aggressive and metastatic. And so the conclusion, P53 mutation status may dictate the extent to which a particular colorectal tumor will be responsive also at a particular molecular targeted therapy, because different mutations are associated with activation of different pathways and different driver mechanisms. Okay, so far for this. Now I want to go, in the time that I have left, to go to the second story. The second story belongs to, again, a very talented PhD student, Sapta Parna Mukherjee, Sapta in short. And Sapta asked or wanted to look for non-transcriptional mechanisms of mutant P53 gain of function. And the approach she took, in this case, she started with PANC1 cells, a common human pancreatic cancer cell line, which also happens to carry the 273H mutation, just like SW40. In this case, single mutation, there's no additional mutation in these cells. And she took those cells, either parental or again, knocked out, CRISPR-Cas9 knocked out for P53, and did mass spec analysis to identify proteins that interact with the mutant P53 protein in these cells. And this is the analysis of mass spec, and P53, of course, came out as the top hit in this immunoprecipitate, as expected. And there were a number of other proteins that were significantly enriched. And one of them that caught our eye was this protein SQSTM1, or sequestrosome 1, better known as P62. And this is just a validation by co-immunoprecipitation, immunoprecipitating either P53 or P62, and you can see robust and very clear specific mutual co-precipitation. In this case, P62 coming down to P53, in this case, P53 coming down to P62. Robust and easily detectable interaction. Okay, and just to validate this by a different method, SAPTA also, and to see where this interaction occurs, SAPTA also performed proximity ligation assay, PLA in short, and these dots are indicate sites of interaction between those two proteins, the distance of less than 40 nanometers. And as you can see, this is control cells, in which in this case it's not knockout, it's cells in which P53 has been knocked down with shRNA. So we have a very small residual amount of mutant P53 still in those cells. These are the control cells that have their normal high amount of stabilized mutant P53. And you can see very prominent interaction between mutant P53 and P62 in the control cells, but not in the P53 deleted cells. So what's P62? P62 is mostly famous for its role in selective autophagy, and it's involved in degradation of aggregates, organelles, and many, many additional things. It is also involved in proteasomal degradation, but it is, I would say, far less studied in this regard. Okay. So the next question that SAPTA asked is this interaction common to all P53 mutants? And she expressed 273 and 175 in parallel in this case, she did it in the cell line, in the lung cancer cell line H1299, but similar experiments were done also colorectal cancer cells and in the pancreatic cancer cells to begin with. And as you can see here, in this example, these cells just are convenient because they are P53 null to begin with. You don't need to manipulate them, and you can introduce into them either 273 or 175, or this is empty vector. And this is IP of P62, and you can see that P53 comes down to P62 when you have the 273 mutant, but not the 175. So again, two different classes of mutants, reminding us of something that we all saw before. 273 also in this case has selective binding with P62. 175, which looks very different and is conformationally altered, isn't interacting with P62. And parenthetically, wild-type P53, which is more similar to this, also interacts with P62, but the interaction is weaker than in the mutant. So we believe that this is an interaction that also is shared by wild-type P53, but the mutation and also the accumulation accentuates this interaction. 175 loses this interaction completely. Okay, so we actually, our collaborators did some docking, some structural modeling using docking algorithms to ask where is the interaction site, where is the predicted interaction site between mutant P53, 273 and P62. And I think, okay, sorry. And the prediction was that the interaction would be mainly within this region of the P53 protein, comprising amino acids 331 to 336. So again, our collaborators in this case, the lab of Daniel Schramek in Toronto, generated a derivative of the 273H mutant carrying also 5 alanine substitutions and residues 331 to 335 of the mutant. And very nicely, so this will be mentioned, called 5A from here on. And very nicely and reassuringly, whereas the full length, which essentially is a single mutant, 273, binds nicely with P62. So this is IP with P62 and Western blot for P53. P62 binds nicely the 273H mutant, but it practically loses its binding to the 5A mutant, which is not higher than the non-specific background. So we have a mutant of mutant P53, which does not bind P62 anymore. And this is, again, validation by PLA, PLA between either the intact 273H mutant and P62 or the 5A mutant thereof and P62. And you can see that abundant interaction with the intact 273 mutant and much attenuated interaction when you mutate those 5 amino acids and prevent the interaction. Okay, so back to function. So this is nice. We have interactions. Does it mean anything? So Sapta did a variety of functional assays, cell line-based assays, to ask, are there any features that are shared between 273 mutant P53 and P62 and are lost upon abrogation of both of them, suggesting that maybe these would be the places where we want to look for the significance of the interaction. And what came out most appealing is the effect on migration. So if one depletes, if Sapta depletes mutant P53 from PANC1 cells and does this kind of wound healing gap closure assay, you see that depletion of mutant P53 attenuates gap closure. This is the control, and gap closure is attenuated. This is also shown here. This is a classical hallmark of mutant P53 gain of function, and it was deeply studied by Karen Wusden and is defined perhaps as the most distinctive feature of mutant P53 gain of function in culture, at least. If one depletes, Sapta depletes, that should be SHP62, sorry, it's knockdown of P62. Again, we have a substantial attenuation of cell migration of gap closure, significantly attenuated. And you can see it in this gap closure assay, and you can see it also in this transwell migration assay, 2 different approaches, same conclusion. Both mutant P53 and P62 are required for maximal migration of those cells. And this is also an invasion assay with similar effects. So both migration and invasion are dependent of those cells, pancreatic cancer cells, are dependent both on mutant P53 or on P62. Okay, that could still be a coincidence. Now, does it have biological in vivo manifestations? So again, taking the same cells, depleting either P53 or P62, and looking at tail vein injection and experimental lung colonization. Control cells are highly metastatic in this type of assay. Depleting either P53, mutant P53 or P62 has a very pronounced significant effect. You might note that the effect of depletion of P62 here is stronger, whereas the effect of depletion of mutant P53 is stronger, which tells us they share common features, but each of them has its own additional features which are not shared. And it also tells us that in vitro cell migration assays are not necessarily a good proxy of in vivo lung colonization assays, which some people would argue are not the best proxy for in vivo metastasis. So not surprising, not as simple as a 1-0 thing, but we have a lot of commonality between these two, mutant P53 and mutant P62. And the question then is, okay, before we go to the question, Sapta had this cute idea, which worked out very well, and asking, okay, we say that mutant P53 promotes migration. We say that P62 promote migration. Why don't we take a look at what happens when cells migrate? And so this is this kind of wound healing assay, scratch assay. So this is the cell monolayer. This is where, right, the gap is, and cells are moving into the gap. And Sapta fixed the cells after 8 hours, so they were just in the middle of moving and closing the gap. And she did a PLA. She fixed the cells and did the PLA on cells in the center of this monolayer and cells within the migrating front. And very nicely, as you can see, the interaction between mutant P53 and P62, which is seen in all the cells, including in the center, is much more abundant, much more robust in the migrating front. In other words, when cells migrate, this interaction is formed much more robustly, or those cells that have a more robust interaction to exist to begin with are the ones that move forward and jump into filling the gap. Both of them would be consistent with the idea that this interaction is functionally associated with increased migration, even though it doesn't prove it formally. Okay, so if now Sapta, okay, let's see where we are. I think we still have a few minutes, yeah? Okay, so if Sapta takes a look at the migration front with a confocal microscopy, something was very clear and apparent. The parental PANC1 cells, so this again is the migration front, the same type of assays you've seen before, taking a gap and taking a picture, but in this case, it's just it's looking at what the cells look like in doing immunohistochemistry, sorry. And you can see that the PANC1 cells migrate in this scattered, individualized migration. You can see big gaps between the cells, pretty much individualized cell migration. On the other hand, if you deplete either mutant P53 or P62, now you have this nice epithelial sheath, and you don't have this individualized migration anymore. These cells still migrate, right? You've seen it before, but now they migrate as an epithelial sheath. And reassuringly, there was this very nice paper by Jacob van Reenen, who did intravital imaging of cell migration, tumor cell migration in pancreatic cancer in the mouse, not in the human, of course. And what he concluded is that, migratory behavior of primary tumor cells of a genetically engineered pancreatic cancer mouse model, and found that these cells migrate with the mesenchymal morphology as single individual cells or collectively as a stream of non-cohesive single motile cells. So the parental PANC1 cells reproduce this in vivo pattern of individualized cell migration, which is dependent on both mutant P53 and P62. You take away either of them, they now revert to this kind of collective cell migration, but actually epithelial sheet slower migration, which in pancreatic cancer is less aggressive. In other types of cancer, like lung cancer, it's a different story. As I said also before, different cancer types, the same mutant would have different implications. It's very much mutant and cancer type context dependent. It's not just one of them alone. Okay, so can we prove that migration is really dependent on this interaction, or at least obtain more robust evidence? So here's one approach. So these are, again, these lung cancer cells, H1299. And we have either, you've seen it already, either the control cells, P53 null, or 273H expressors, or 175 expressors, right? This binds P62, this doesn't bind P62. And if one does this scratch, right? Scratch assay, wound healing, gap closure assay, as expected. So these are the knockouts. This is HCT116 cells, wild type cells, P53 knockout. Sorry about this conjunction. P53 CRISPR knockout, reconstituted with 273H or 175H, and you can see that compared to the wild type cells, or to the P53 knockout cells, which are the ones that we really should be comparing to argue gain of function. The 273H cells migrate more, this difference is significant. That 175H cells also migrate more. And this is an agreement with what has already been published by a number of researchers that both those mutants can enhance cell migration. But what happens if now we deplete P62? No effect on the migration of the knockout cells. No mutant P53 there. No effect on the migration of 175 cells. Migration of 175H cells is not dependent on P62. Some other mechanism, whatever it is, we still don't know because we have not looked into it any further. But migration of 273H is totally ablated. So the migration of these cells with these mutant, but not this mutant, is completely dependent, the enhanced migration, not the basal migration, but the enhanced migration, the gain of function effect is completely dependent on the presence of P62. Different modes of migration, different mechanisms underlying them. Okay, so I think we're approaching the end. I try to rush up because I want to give you the bottom line. So Sapta asked, what's the mechanism for this selective migration driven by, or selective effects driven by this interaction, presumably between mutant P53, 273 and P62. And to this end, she did a total proteome analysis. She also did other analysis, but this is the one that was most revealing. And what she found is that when she silences either mutant P53 or P62, some proteins increase in abundance, some proteins decrease in abundance, and most notably, 25 proteins were increased in abundance when either mutant P53 or P62 was deleted. Now, deleting P62 is expected to increase the abundance of many proteins because P62 is a key player in autophagy, selective autophagy, also in PANC1 cells. So presumably most of those proteins are clients of autophagy, and indeed, if you look at them, they are classical. Many of them are classical autophagy cargo. But what was more interesting is, of course, this group of proteins that are jointly upregulated when you reduce either mutant P53 or P62, suggesting that both mutant P53 and P62 normally reduce the expression, reduce the levels of those proteins. Now, without getting into the details, because save time, RNA of those proteins, the ones that we checked, and also comparing to RNA-seq of the same cells, is not altered. So we are talking about something which is not transcriptional, but rather post-transcriptional, presumably post-translational. And interestingly, when Sapta looked at the list of those 25 proteins, what emerged as a significant component was a variety of proteins involved in cell adhesion. And this is an example of one of them, connexin 43, which is a gap junction protein. It was one of the top hits. And as you can see in this staining, in control cells, PANC1 cells, you can also notice connexin 43 is a gap junction protein. But you would notice that, in fact, in these cells, it has a unique localization. It's not in the gap junctions. In fact, these cells are scattered and they don't really have gap junctions. And it has this localization, which again, is of interest. But if you deplete either P53, mutant P53 or P62, what you see now is that, first of all, you have more of this protein. You have more connexin 43, more pronounced in the case of P62, less pronounced in the case of mutant P53, but in both cases you have an increase, and more importantly, now you see gap junctions and you see connexin 43 in the gap junctions, which would imply that the presence of mutant P53 and of P62 causes rapid degradation of this gap junction protein in several additional cell adhesion proteins, and presumably as a consequence, these cells don't have gap junctions, don't have cell adherence junctions, and in fact migrate as scattered cells individually rather than stay as a model, as an epithelial sheath. Okay, so I think for the sake of time, I won't get into, I'll just show briefly. So what happens in the normal cells? Normal, these are cancer cells, right? In the non-manipulated cells, where's connexin 43? It's here. Very weird localization. And without getting into all the details, this turns out to be the Golgi based on staining with different markers. So in these cells, connexin 43 ends up in the Golgi and does not get to the cell junctions. And in fact, and I think that would be the last one. No, I go to let's skip it. Go to this one. Okay. What SAPTA could show is that mutant P53 and P62 promote the rapid degradation of connexin 43 and of additional cell junction proteins in the proteasome when they are present together. So maybe I'll just try to dwell on that, and it's a little busy, but not very, very complicated. So these are the control cells having mutant P53 and P62. If SAPTA inhibits the proteasome, we get massive accumulation of connexin 43, telling us that in these cells, connexin 43 is primarily degraded via the proteasome. However, if she inhibits the lysosome with chloroquine, right, inhibition of autophagy, autophagic degradation, we also get a robust increase, although less than here, telling us that a substantial fraction of this protein is also degraded via the lysosome, presumably via autophagy. And this has already been reported before, that connexin 43 is a client of autophagy. But as we see, it's even more in these cells, it's even more a client of proteasomal degradation. If you now deplete mutant P53, what happens? MG132 inhibition of the proteasome doesn't do anything, telling us that if you deplete mutant P53, there is no more any proteasomal degradation of connexin 43, telling us that mutant P53 is obligatory for the proteasomal degradation of connexin 43. On the other hand, if you deplete P62, what you see is that, first of all, in P62, you get a huge amount of connexin 43 accumulated. You also saw it in the staining. And neither proteasomal inhibition, nor chloroquine, nor lysosomal inhibition has any further effect, telling us that P62 is important in these cells for lysosomal degradation of connexin 43, right? Which is not surprising, it's a well-known fact, but is also obligatory for the proteasomal degradation of connexin 43 because no P62, no proteasomal degradation. So I think I want to jump to the conclusions because I think we have to get to the end. Yes, we have to get to the end. So I'll just get to the model of what we believe we know now. And that is that binding of mutant P53 to 73H and not 173H to P63 augments the ability of P63 to bind and promote the proteasomal degradation of a subset of ubiquitinated proteins, which include cell adhesion proteins. And this enables the cells to detach from the epithelial sheath, resulting in individualized cell migration and cancer cell spreading. Okay, I think I will stop here and acknowledge the members of our lab. And just to point again, this is SAPTA, who did the non-transcriptional mechanism study. And Ori is hiding somewhere. Ori is hiding here, not really hiding. This is Ori, the first story. And these are our collaborators. And while you read the list of the collaborators, I would like to thank you. And I'll be happy to answer questions.
Great. Thank you so much, Moshe. Really fascinating work. We have two questions. If anybody else would like to ask a question, please type it into the Q&A, and I'll be happy to read them off. The first question from Irina: what are the molecular mechanisms by which R273H mutant P53 protein is recruited to the regulatory elements of the signature genes?
Okay, great question. We are still working hard to figure it out. We know that these sites are GC rich, so presumably, and they're predicted to be binding sites of transcription factors that are binding GC rich elements such as SP1, for instance, which has been reported to interact with mutant P53, but so far by depleting only single transcription factors, we still don't abrogate the activity of 273. So we think either it's a GC-rich binding protein that we have not tested yet, or that it could be, alternatively, there could be more than one protein that brings the 273 specifically to these sites. But I would say it's still mechanistically, we still have a way to go. I think one interesting observation that we noted, maybe not very surprising, is that in cells which express 273, the regions to which it binds are less the DNA of those regions is less methylated than in cells that express 175. So there is an involvement of DNA methylation, but it could be the chicken or the egg, and of course, one would have to test it directly.
Great, thank you. The next question is a technical one. Erica asks, which program do you use to quantify PLA signals?
Oh, I should ask Sapta. I'm embarrassed to say that I don't know, but I'll be happy to ask her and send all the information.
That's fine. That's fine.
I think no. I don't know. I know that she was using Imaris, but I don't want to say that for sure. I can check, and if you send me the email, I will be happy to ask Sapta to send back the answer with all the details.
Okay, Claudia, maybe you can find it. Erica's email. The next question is from Jonathan. What is the role of gap junction degradation in healthy adult tissue? It's a very good question. I'm embarrassed to say that I don't know for sure. Gap junctions are degraded and recycled essentially in tissues, in cells which are moving. So they are recycled, they are reassembled in cells which are in a normal layer with cells when they're not moving. The half-life of gap junction proteins, as far as I know, is pretty high. And in fact, it is there to keep the cells together. They probably are recycled like any kind of memory proteins. They're not sitting there forever. Even if the cells are not multiplying, there is some recycling. There's always some production, some recycling. But my understanding is that the overall rate of turnover is not high in stationary and non-dividing epithelial cells. And it's just more probably maintenance, quality control, and some level of tear and wear. But they are believed to be relatively long-lived proteins. Okay, we're just about out of time. Well, really quickly, one last question from Sarah. If R175 mutation is important for initiation of colorectal tumors and 273 is important for progression or metastasis, do many tumors end up with one allele of each, or do you think that an early 175 limits the potential for progression? We would have loved to, I would have loved to say yes, because that would be perfect. We looked at it and it's not as simple as that. So very much, I would say, I'll give one answer because one can give many answers. There's now growing evidence from many labs, including labs in your geographical location, that actually normal tumors, especially normal tissues, especially normal tissues of individuals while they are aging, acquire P53 mutations and many other mutations. And these mutations are just sitting there. Maybe they are doing something very locally for modest clonal expansion, but they are definitely not driving cancer. But then when additional events occur, and we know that P53 mutations are not initiating events in colorectal cancer, they are progression events. So depending on what mutations have been already sitting in this cell, the course of the disease might be different. So what I would propose is it's not that the 175 is driving beta-catenin pathway activation, Wnt pathway activation, but once you have a Wnt pathway activation, having a pre-existing 175 mutation suddenly becomes an advantage.
because in some way, probably these things are compatible with each other. Having a 273 doesn't help maybe, or helps less, so that it is not enriched as much as 175 in the low stage tumors. But if the tumor happens to have a 273 mutation, it will run more fast and more likely towards a more aggressive state. I think that's speculation which I would favor, but there could be other explanations as well.
Excellent. Great. Well, thank you so much for a fantastic talk. Very provocative, lots of cool questions. We really appreciate it. And I think with that, we're going to end. And thank you, Moshe. Thank you for the audience. Everybody have a good day and happy holidays.
Thank you very much. And wishing you all happy holidays. And hopefully what we all are wishing to ourselves, and we all know what it is, we very much hope that will happen within 2022. And we'll be there eventually, where we wanted to be in 2021.
Agreed. Take care.